enormous experience in TB control. For over 18 years, he has been working in the Americas, Africa, Europe, and Asia on TB laboratory strengthening activities. Dr. Scholten has worked at the New York City Department of Health TB control program, also at the WHO European Region TB program, and is currently with the KNCV Tuberculosis Foundation. At KNCV, Dr. Scholten is the chair of the KNCV Laboratory and Diagnostic Strengthening Thematic Group and leads activities with strategic focus towards laboratory and diagnostic strengthening. While working for WHO, he was a founding member of the WHO European Region Laboratory Strengthening Task Force. Okay, so now I would like to give the floor to Jerod.
Hello, everyone. I wanted to welcome you to this seminar. I hope that it's useful for you. I am sitting here with my colleague, Dr. Jacques van den Broek. He's also an expert, a clinical expert in MDR-TB and a GLC consultant for KNCV. And he will, if needed, assist me with the question and answer period, and he's coordinating this from the KNCV side. I will turn off the video now to save space, and I will continue with the talk. Thank you. Okay, so just to give you some background on KNCV, our organization has existed for over 100 years and is exclusively dedicated to TB control. We work in over 40 countries worldwide and work to capacitate national TB programs to control TB with the best possible tools. We are also the Secretariat for the TB Care 1 Coalition, which is a working partner with the TB Care 2 Coalition, which includes Partners in Health. Both are generously supported by the United States Agency for International Development. We would like to thank the MDR-TB Training Network and Partners in Health for inviting us to participate in this important series of webinars. KNCV considers the timely detection and treatment of drug-resistant TB as a high priority. As Dr. Golubkov mentioned, I will now give a brief talk about laboratory strengthening in the context of drug resistance diagnosis and monitoring in low and middle income countries, followed by some quiz questions and discussion. So the next slide is here. Okay. Oh, where is it? So, my presentation will focus on three areas. First, the current laboratory preparedness for drug resistance diagnostics. Second, positioning, or in other words, the functional purpose of a lab network in terms of programmatic management of drug-resistant TB. And thirdly, laboratory network requirements, both ideal and minimum on the national and local levels. This is not working. Okay. Maybe. Okay. As Dr. Golubkov mentioned earlier, WHO recommends that 100% of previously treated TB patients are tested for MDR-TB because retreatment cases are much more likely to have developed drug resistance than new patients. Some of these retreatment patients may be at higher risk, however, for MDR-TB than others. Retreatment cases include so-called chronics, those treated more than once previously for TB, failures of previous treatment who are at high risk for MDR-TB, particularly those who have failed the Category 2 WHO standardized retreatment regimen, relapses, and return after defaults. It is also a global Stop TB target to test 20% of new TB patients. Known contexts to TB MDR-TB are considered at high risk for MDR-TB, as are healthcare workers with TB and TB patients who do not convert their smear sputum status at or after 2 months of treatment. By 2011, 21 of 36 countries with either a high burden of TB or MDR-TB had at least one laboratory capable of performing culture for tuberculosis per 5 million population. In most of the high-burden countries in Africa and the Indian subcontinent, the coverage is particularly low, while conversely, many of the countries in Eastern Europe are over-provided, which raises concerns about the quality of the tests performed. Overall, for all regions in 2011, DST coverage was higher among retreatment cases than among new cases, but at far less than 10%, this is hardly the 100% overall coverage target. In the European region, while the coverage in new cases exceeded the 20% level targeted, I will use this. Incomplete reporting on certain categories of retreatment status is common in the Russian Federation and elsewhere, and as a result, the overall coverage among retreatment cases is low. The lowest coverage was seen in Africa and in the Southeast Asia region. Second-line DST, as Dr. Golokov also mentioned, is recommended for all confirmed MDR-TB patients. In 2011, only 23% of MDR-TB patients were reported with a test result, again far from the 100% expected. The high coverage in Africa drops to 9% without data from South Africa included in this slide, and the proportion of cases tested for MDR-TB in Africa from the previous slide is still very low, so this graph may be a bit deceptive. Underreporting of test results accounts for a certain degree of low coverage in certain regions, while many simply are not tested at all. So with the exception of the former Soviet Union, historically low and middle income country laboratory networks have prioritized detection of smear positive cases as per former WHO recommendations, and therefore they are not designed to detect drug-resistant TB. So these countries need to retool, or in other words, redesign their laboratory networks. All low and middle income countries need to have capacity for accurate and timely diagnosis of MDR and XDR TB. Smear microscopy has been around for well over 100 years and is still the primary means of diagnosing TB in most countries, and can be effectively used as the first screening tool for TB suspects. It also has the advantage of being inexpensive, can be placed at the clinic level, and can rapidly detect infectious TB patients. It can also be used to monitor patients during their treatment to see if they're getting better, such as becoming smear negative within 2 months of treatment. It requires limited training, has a low biosafety risk, and it has the advantage that one can quantify the number of bacilli in the sample, such as few bacilli to numerous, or in technical terms, scanty versus 1+, 2+, 3+, depending on the magnification field used of the microscope and what's observed under the microscope. In terms of PMDT, the big problem with smear microscopy is that it cannot distinguish MDR-TB from non-MDR-TB. It also has a low sensitivity compared to culture and to rapid novel molecular WHO recommended methods, which I will describe shortly. Light-emitting diode fluorescent microscopy, which was endorsed by WHO within the last few years, is recommended by WHO to completely replace the traditional light microscopy that was discovered over 100 years ago, as LED fluorescent microscopy has 10 to 20% higher sensitivity than the traditional light microscopy. But LED fluorescent microscopy is still very insensitive compared to culture and the novel molecular methods. Conventional culture is considered the gold standard for first and second line DST. Liquid culture with BACTEC midget is slightly more sensitive than conventional or so-called solid culture. Both methods can detect MDR-TB from any part of the body and are highly sensitive and specific to detect TB. Methods to conduct both methods are well established and agreed upon. The most reliable and reproducible first-line drugs for testing are INH and rifampicin, isoniazid and rifampicin, whereas the second-line drugs that are most reliable and reproducible for testing are fluoroquinolones and second-line injectables. Patients under TB treatment can have both live and dead bacilli in their samples that they produce. Conventional culture detects live MTB bacilli, which can be used as an important clinical indicator for monitoring of patients' treatment. The big disadvantage of conventional culture in DST is that it requires intensive resources. Because of the complicated inputs needed for culture in DST, the cost is also relatively high compared to smear. In low and middle income countries, culture in DST cannot be placed beyond the intermediate level and are often only at the reference level laboratory in the capitals of countries. The biosafety risk to laboratory workers is also high with culture and DST. The live concentrated bacilli, if not disposed of properly, can also be a risk to the general public. It is accepted that the contamination rate for culture and DST is between 5 and 10%, with a slightly higher contamination for liquid culture. So patients with contaminated cultures need to have repeat tests. This can be a problem given that the patient may have already started treatment, and it is best that the culture in DST for diagnostic purposes is performed before a patient starts anti-tuberculosis treatment. The results for DST, first-line DST, take 3 to 12 weeks, with liquid culture taking the least time and conventional culture taking the longest time. Line probe assays, also called the Hain test commercially, have been approved by WHO to rapidly detect INH and rifampicin resistance with molecular methods. They are highly sensitive and specific. In 2011, of 36 high TB and/or high MDR-TB burden countries, 9 of them, or 25%, reported that they had at least one line probe assay laboratory per 5 million population. These tests can also be used on confirmed sputum smear positive TB cases to test for MDR-TB rapidly, or to further validate expert MTB RIF positive cases. I will describe subsequently expert MTB RIF testing. The disadvantage of these tests is that they are only recommended for sputum smear positive cases or culture positive cases. They are not currently endorsed by WHO to detect second-line drug resistance, although a kit is available for this. They need fairly sophisticated labs with lots of space to avoid contamination of the samples. The cost is higher than smear, but slightly less than culture and DST. Highly trained technicians are needed, but it is slightly less complicated than conventional culture and DST. It is not suitable to be placed on the district or clinic level, and there is a moderate to high biosafety risk to laboratory workers. The risk is lowest when dealing with only processed sputum and higher when culturing. A lot of you may have already heard about expert MTB RIF. It can detect TB and RIF resistance as a surrogate marker for MDR-TB within approximately 2 hours. It is highly sensitive and specific to detect TB, similar to conventional solid culture, but not quite as sensitive. It can be placed at district and sub-district levels, unlike any of the other WHO recommended tests I have described besides smear so far. It also requires limited training and has a low biosafety risk comparable with smear microscopy. While at KNCV, we think that this is a breakthrough for TB diagnostics and MDR-TB diagnostics. It is not a perfect test. It can only be used for sputum similar to line probe assays, so extrapulmonary cases can only be detected with conventional culture and DST. It does not detect INH resistance. While in most countries RIF resistance is associated with INH resistance, some cases can be mono-RIF resistant and susceptible to INH, which is one of our most powerful drugs to fight TB. In terms of detection of RIF resistance, as with any diagnostic test, the positive predictive value of the test is lower when the prevalence of MDR-TB in a population is lower. So for instance, if the prevalence of MDR-TB in the population tested is around 2%, approximately half of the expert RIF positive cases will be false positives. When the prevalence of MDR-TB in a population is 15% or greater, however, the false positivity rate is very minimal. Another problem is that it is not really what we would consider a point-of-care test done by the clinician quickly at the bedsides or the bench, in the diagnostic room, such as with a dipstick. It has some infrastructure requirements because it is electronic and has single-use cartridges with reagents inside. These cannot exceed 30 degrees Celsius, neither the machine nor the cartridges. It also needs a stable supply of electricity, which is a challenge in many low and middle income countries. Each cartridge costs about $8 US, which is after a buy down by major TB partners with the manufacturer to make it more affordable for countries. It also requires annual calibration of the machine, which has cost and logistical implications. Just to give you an idea of what the process and machine look like if you haven't seen it before, you take a sputum sample and mix this with a reagent that comes with the kit specific to that cartridge, and you process this in a plastic cartridge. That hands-on work takes about 30 to 45 minutes. Then you place the cartridge into the machine and it is processed within 90 minutes. The results tells you with a computer screen whether TB was detected or not, and if TB was detected, if RIF resistance was detected or not. The result is also printable and you have a database associated with the machine. WHO estimates that expert MTB RIF could increase the diagnosis of TB patients with drug resistance threefold by 3 times the current level. However, similar to culture and DST and LPA's line probe assays, things can still go wrong with expert, and this estimate may be overly optimistic at this stage. For instance, some countries we work with have ended up with cartridge shortages or expired cartridges due to chronic system-wide supply management issues. In some countries, all patients are not put on empirical treatment for MDR-TB with an expert RIF positive result until conventional culture and DST are confirmed. Others do not have timely reporting of results back to clinicians, so the value of expert TB becomes less in that situation. By the end of June 2012, 67 low and middle income countries had started using expert MTB RIF testing. Just to summarize, solid culture and DST are reliable, but the traditional process takes 9 to 12 weeks for a first-line DST result. Liquid culture and DST take significantly shorter time with 3 to 5 weeks, and for that reason WHO has recommended liquid methods as the gold standard in culture and DST. Line probe assays after smear microscopy of sputum is confirmed can detect MDR-TB, both INH and RIF resistance, within 1 to 2 days. And finally, expert MTB can detect on pulmonary specimens a surrogate marker for MDR-TB, namely RIF resistance, within approximately 2 hours, closer to the patient than any of the other methods that I've described. Overall, the positioning or functional purpose of laboratory services for PMDT should be considered for both initial diagnosis and for treatment monitoring after diagnosis. For diagnosis, it would be ideal to have rapid diagnostics in most low and middle income settings to enhance the timeliness and coverage of MDR-TB detection, which, as I showed earlier, remains very, very low with the current infrastructure utilized. Expert MTB RIF may be used as an initial test for MDR-TB suspects replacing smear microscopy. In many countries, the prevalence of MDR-TB among retreatment patients or certain categories of these patients is quite high, as Dr. Golubkov has already mentioned. In the parts of the Russian Federation, it may be 40% by now for new patients. But for retreatment patients, it's often 10% or higher in a lot of countries, besides in Eastern Europe. Although in some countries we do not have a clear recent picture of drug resistance, as we've seen also from Dr. Golubkov, it is recommended that in general retreatment patients can be put on empirical MDR-TB treatment. In some countries, new patients might also be put on empirical MDR-TB treatment, particularly in Eastern Europe, where the prevalence is high among new patients. To speed up confirmation and not delay diagnostics, LPAs and conventional culture and DST can also be used, and the results of the conventional methods, preferably liquid culture due to the speed, can guide clinical decisions for treatment. In terms of treatment monitoring, MDR-TB patients on treatment should be monitored monthly with both smear and conventional culture, at least until they're culture negative after 3 consecutive cultures. Molecular methods, including expert MTB and line probe assays, are not recommended for treatment monitoring by the World Health Organization. In terms of laboratory network requirements for PMDT, it would be ideal to have a combination of multiple tools for diagnostics and monitoring, including smear microscopy, preferably LED fluorescent, conventional culture with first and second line DST, again preferably liquid due to rapidity, plus rapid WHO-endorsed diagnostic tools such as expert and/or line probe assays. It is also necessary for all countries to have a linkage with a supranational reference laboratory for quality assurance of culture and DST, and if it cannot be performed in the country, then outsourcing via the supranational reference laboratory all second-line testing for all laboratory-confirmed MDR-TB patients. On the national level, because the ideal is not always realistic, but is something we should strive for, the minimum requirements for a country to have appropriate PMDT laboratory infrastructure include 1 microscopy laboratory per 50 to 150,000 population, 1 culture and first-line DST laboratory per 5 million population, second-line DST capacity available either in the country or outsourced to another country, and a formal linkage with a supranational reference laboratory for quality assurance and, if needed, for second-line DST. At the level of the district or periphery PMD center, there are 2 distinct minimal model standards needed. The first model standard, based on conventional methods only, namely Model 1 here, has the following minimum requirements: smear microscopy be capacity at the PMD center or very nearby, reliable and rapid sputum sample transport network to the laboratories where culture and first-line DST can be done, and a mechanism for internally or externally testing second-line DST. It should also have a well-functioning reporting of results by email, for instance, shared database, ETB manager, cell phone. If the results are not reported back to the clinician, doing the test is useless. The second minimum model standard combines conventional and rapid diagnostics, namely Model 2, and this can include rapid diagnostics at the district peripheral level with expert MTB and or line probe assays on a higher administrative level, and all of the features from Model 1. However, with Model 2, the volume of smear, culture, and first-line DST might be reduced with the widespread introduction of expert MTB, RIF, and LPAs, so the burden on the central, or in some cases regional, culture and DST laboratories would be reduced. This is just a slide to give you an idea of the supranational reference laboratory network which each country should affiliate itself with. The current coordinating laboratory for the entire network is in Antwerp, Belgium. They also ensure that the other supranational reference laboratories are performing adequately to support countries. Currently, there are 28 supranational reference laboratories. 4 additional supranational reference laboratories are being considered to be added to the network, including Benin, Uganda, Pakistan, and Denmark. While I have just presented 2 standard model possibilities for lab requirements for PMDT, there is no one-size-fits-all solution in reality. The solution that a country comes up with should be a consultative process based on what works to most rapidly and accurately diagnose MDR-TB patients in that setting. Consideration should also be considered for the burden of MDR-TB. If a country has a high burden of MDR-TB, they may have a really hard time achieving 100% coverage for all retreatment patients and selected new patients with conventional methods. And perhaps Model 2 would be a better option for them. If, on the other hand, the current infrastructure and burden of MDR-TB can be handled with conventional methods as described in Model 1, then the value of introducing the novel molecular WHO methods would have less of an impact in that setting, particularly if culture and DST labs are considered well established and quality assured by the supranational reference laboratory. They have a secure, stable budget line without service interruptions and well-trained staff with low turnover. The approach should also depend on what PMD program models is being utilized or envisioned for the future. As Dr. Golubkov again mentioned, generally WHO recommends that PMDT programs are decentralized or based on a community-based model. But in some countries, they still have these centralized models with laboratory services located centrally. And if there are a few patients, the distances are not so large within the country, and there's a reliable and rapid transport network, that approach could be still feasible for certain countries. So when a country or a PMD program is considering how to structure or restructure laboratory services, the following technical and programmatic considerations should be seriously considered. First of all, you want to make sure that your laboratory staff and the general public are safe from the methods. So you have to seriously consider biosafety. You also have to consider the infrastructure for both the equipment function and biosafety, with some higher technology and higher biosafety risk technologies such as cultured DST and line probe assays. You would need biosafety cabinets and a containment laboratory. You also have to consider the cost. There are some cost-effectiveness studies that go into this. But I like to always say that, The HIV/AIDS programs have really been better advocates for laboratory strengthening in recent years than the TB world, and I think that we can use them as a model. They took these fairly sophisticated tests for viral load testing and CD4 counts, and were able to get those out into the field in a very standard fashion and efficiently, and I think that we can learn from that. And so we shouldn't always worry about cost. We have to be reality-based, but maybe something is going to cost a few more dollars, but it has a much bigger impact. We need to think about that. Human resources are a big consideration. If you have a population that has, or an infrastructure that has a high turnover of staff, and you train these highly technical people for a year to learn how to do culture and DST, and they leave 2 months later and you have to repeat the whole process again, that's a problem. And you need, maybe a different model needs to be considered if that happens repeatedly. You depend less on the highly technical laboratory methods. Recording and reporting are also very important. You need to link the laboratory results to treatment decisions and to ongoing surveillance. Management of lab commodities and supplies is very important. We often, in the countries that we work, find that there's something missing, some reagent missing, that completely paralyzes, or the cartridges or whatever, that paralyzes the services. And then you have to ensure that the tests that are being done have are quality assured, externally and internally quality assured, and that the laboratories are supervised and monitored appropriately. Again, any disruption in any of the above that I described will likely result in suboptimal detection and clinical monitoring of MDR-TB cases. A major issue for countries is the transport of patient samples either within the country or internationally. Biosafety has to be considered. The biosafety risk for sputum is quite low, whereas high for live culture specimens. The viability of specimen also has to be considered. If stored too long in excessive heat, TB bacilli decompose or denature and become unlikely to be able to be cultured and DST'd after 3 days. If a transport network cannot get specimens to a lab within 3 days, preservatives like CPC can be considered to reduce the denaturing process. GeneXpert and LPAs are molecular tests, and they are less sensitive to the denaturing process, but DNA still degrades, and it can eventually affect the results of those tests also. International standards for transport of samples can be very strict, and there are special trainings offered to assist countries in legal and procedural issues to facilitate international transport, which I won't go into here. From the patient perspective, which we should never forget, we need diagnostic and monitoring methods that are most convenient for them. This includes low or preferably zero cost, nearby diagnostic capacity, very accurate tests so that we don't tell them one day that they have TB and the next day they don't, or we treat them for MDR-TB mistakenly, or vice versa. The number of tests, every time you ask them to do a test, they have to produce something from their body, and that can be invasive and discouraging. We can lose them. They can default or die in the meantime. And the timeliness of tests. We need to make sure that they're very rapid tests. In order to rapidly detect, diagnose, and treat M and XDR-TB patients, the world needs to rapidly increase the access and availability of diagnostic tests that are accurate, cheap, fast to use, require minimum training, need simple infrastructure, and are based on both sputum and non-sputum samples. In order to monitor treatment with new novel methods, distinguishing from dead and live TB bacilli and quantification of concentration of bacilli in samples tested would also be useful. This graph I find which indicates by distance from patient and time the various WHO-approved technologies and expected tests in the pipeline. As we do not have time remaining, I would just like to summarize that we are expecting another rapid test that is closer to the patient than GeneXpert MTB RIF by 2015. Okay, I hope that we have time now to move on to the quizzes.
Jerod, would you mind to read the question for everybody?
Sorry, Dr. Golokova. The two novel methods described in this talk, including line probe assays and expert MTB RIF testing, are approved by the World Health Organization to detect pulmonary multidrug-resistant tuberculosis. True or false? Or neither true or false? Okay, it looks like the voting is stopped. Okay, the answer is it is false. Most of you, 76.4%, got that correct. WHO only recommends these tests to be used for pulmonary TB, to detect pulmonary MDR-TB. Okay, the second quiz is, which of the following is the best description of the ideal use of existing technologies for the detection of MDR-TB as currently recommended by the World Health Organization? Number one, smear microscopy can be used to diagnose all forms of TB. 2, conventional culture and DST, including solid and liquid, can detect all forms of TB rapidly and with limited investment. 3, expert MTB RIF tests can rapidly detect all forms of TB and can completely replace both conventional smear and culture methods. Or 4, along with appropriate infrastructure, human resources coordination, and quality management, a combination of conventional and novel methods can ensure a higher quality and more rapid diagnosis than any WHO recommended diagnostic technique alone. Okay, yes, most of you, even more, got this right. The correct answer is 4. 82% of you, 32 participants answered that correctly. So we can go to the next quiz if we have time. Okay, so this one is, which of the following is not true? 1, expert MTB RIF testing may be utilized to start treatment for MDR-TB within 2 hours after testing as a first diagnostic test for MDR-TB suspects. 2, new patients should never be offered empirical MDR treatment based on expert MTB RIF testing. 3, line probe assays can further validate expert MTB RIF testing results. Or 4, conventional culture and DST remain the current gold standard for detection of MDR-TB, and the results, if and when available for a particular patient, should ultimately guide clinical decisions for MDR-TB treatment. So maybe we have time for another?
No, Jared, we don't have time for quizzes. We have few questions for you, actually about 10, 15. So we'd like to use it sometime before we close for questions, if you don't mind. So now you can see questions, you can read them and answer when you feel comfortable.
Yeah. Okay, we'll start answering some of these questions with the time that we have. One question was, what will the utility of AFB smears in settings where TB is endemic, but availability of quinolones is also endemic over the counter? As I think we are missing 70% of TB patients by smear negatives and use of quinolones. That will not change anything. You should still use smears. There will be the use of quinolones won't change anything in terms of detection with smears, but there's still a use for smears because you'd still be able to detect TB, and then you'd have to determine whether it's MDR-TB. Okay. The shape of AFB under microscope, say drug-exposed or drug-resistant, for example, between the normal rod shape and the distorted ones. No, that doesn't change. The shape doesn't change. Which factors can influence a high contamination rate and be checked retrospectively for possible reasons of contaminations? Patient may blame health workers and vice versa. There are specific factors that laboratory consultants use to evaluate the contamination rate. It can be related to, as I mentioned, the transport, that the viability of the sample was arrived at the lab in such a condition that it was more likely to be contaminated. But there are also laboratory techniques that can lead to contamination. You can cross-contaminate is one issue, and then you can cross-contaminate one patient sample with another by reusing a pipette or something, and then placing that in another sample. Or you can have some environmental contaminations from water, for instance. So there are ways of retrospectively looking at those, and WHO and CDC have some guidelines on that, as does TB-CAP if you go on the KNCV website and under TB-CAP or TB care and look under lab tools, there are some SOPs on culture and how to look at this. Okay. Okay. Is it only WHO endorsed for sputum? Can other samples be processed by Expert MTB RIF? Expert MTB only works with sputum samples or can use for other non-pulmonary TB? Okay. That's also a burning question. There is some evidence that Expert TB does work for certain types of non-sputum samples. For instance, gastric aspirates, which are important for diagnosing pediatric TB. But the WHO has not yet has not convened to look at the available evidence to be able to recommend that non-sputum samples are used. Some samples are less promising. For instance, brain fluid or meninges fluid. It looks like that is not very sensitive to the expert. Okay. How long will MDR-TB be detected in expert after TB treatment full course? That's a good question, and I'm not sure, but it depends. If the treatment is successful, what the expert is detecting is bacilli, either live or dead, in one's body. So, and even after usually 2 weeks of effective treatment, you no longer have viable, between 2 weeks. For MDR, it can be slightly longer, several months. But at some point, you should not have any live bacilli, but your lungs still are clearing the dead bacilli. But by the end of a full course of effective treatment, you shouldn't have a positive expert test. So, but I guess you were implying that you might be able to use that for monitoring also, similar to smear microscopy, which is not yet recommended from WHO, if it ever will be. Okay. Is this ETR? Okay. Okay, I guess everyone sees these questions. I'm looking at, we have LPA and expert, and it's difficult for the clinician to choose between them for the new cases. So we start empirical treatment with RIF resistance, but actually we could also check for INH at the same moment, which is economically more efficient. It's not just economics, it's also effectiveness. Because LPAs cannot be located on the peripheral level or the subdistrict level like expert, and if you don't have a good communication network or a good transport network, then you're probably better off using expert. But if you can ensure that the Hain test will be used properly and you'll be able to transport the sputums and communicate the results back quickly, and have the infrastructure for the Hain's test and the higher staff trained, then that's also an option. So you have to consider those, and there's no, like I said, there's no one-size-fits-all situation. You have to think about the setting. And you also have to think about the MDR-TB prevalence. So, because as I said, in some cases, new cases might, if the prevalence among new cases for MDR-TB in the general population is quite high, then your empirical treatment of the new cases would even, it would be highly likely that it would be MDR-TB anyway. So. Maybe with time we stop performing LPA. I'm not exactly sure what you mean, but I think over time, it's possible, we're hoping that we'll have even better tools. Yeah. Yeah. So, so there should be newer models out that are improving. So I think that we have made leaps and bounds, but we still can fine-tune these tests. The ideal test for fluoroquinolone resistance right now is the recommended one is using conventional methods, as I mentioned, but an ideal one would be if we could get something close to the patient, you know, that was just, it told us the whole, like GeneXpert, but it told us the whole range of all the drug resistance possibilities, you know. That would be perfect, you know. In 2 hours we know the whole spectrum of drug resistance. For me that would be ideal, or even closer to the patient, a dipstick that tells us that. Okay. So pointing towards cost, can we decrease the number of cultures in early converters from monthly to alternate months after, say, 2 to 3 negative cultures? Yeah, I think that my expert colleague says not in an initial phase. I think that you can go in and out, being culture positive and culture negative for various reasons during MDR-TB treatment. And so it's important that you have constant monitoring in the early stages. Okay, I think that we've answered most of the questions here. Are there more questions?
No, that's most of the questions, Jerod, we have, and unfortunately we have about 2 minutes to finish.
Okay.
So yeah, so I would like to thank you, Jerod, very much for very interesting and informative talk, and I would like to also thank you